Hello, I will present you today the Qiime-2 analysis pipeline, and this is my summer project. And this video is made for my supervisor, Professor Osmay Güre. For my outline of the pipeline, my project, I will be talking about a brief introduction about the topics that I will mention, such as 16sRNA, Earth Microbiome project, and sequencing platforms. And then I will mention an example experiment that I have been done in my report. The experiment is about testing the hypothesis of whether the microbiome in the mice contributes to the development of Parkinson's disease or Parkinson's disease influenced the fecal microbial community diversity. The analysis of this experiment and the basic introduction for my report will be done. Then, roughly, I will be talking about what have been done for my 3 experiments in my report. First, we will import the files into the program, then demultiplex these sequences, pick the ACVs, I mean amplicon sequence variances, and then generating a phylogenetic tree. After that, to doing an alpha verification and choosing sampling depth. For using all of three things, we will do a core metrics results analysis. These will be given us alpha diversity, beta diversity, emperor analysis, and PCOA plots. And we will use them for analysis, and then we will assign them taxonomically. For taxonomic assignment, we will also train a classifier. After that, we will do abundance testing, abundance analysis with using ANCOM, and then we will do time relative change analysis, which is another name, longitudinal analysis. And after that, we will conclude our analysis with meaningful words. Okay. So let's begin. What is amplicon? Amplicon is basically a result of an application process or replication process composed of DNA or RNA. It's just basically a PCR product. You can also say that. For my analysis, it's the product of the Illumina sequencing protocol. In the genome, there are actually, in the genome of the species in a domain, there are conserved regions. These regions are present in every species in that domain. But for phylogenetic marker regions, these conserved regions are for bacteria, 16sRNA, also archaea, and for archaea and bacteria, more like archaea, 18sRNA, and for the ITS DNA, it's the fungi. These regions actually basically conserved, but they have specific regions that differentiate between the species. And for this species, these regions are conserved with having the hypervariable regions inside them to differentiate between the species inside the domain. For this analysis, it has been used the bacterial species and hypervariable region 4, which is right here. How it's done is basically putting a forward primer at the beginning of the known sequence in the 5' and reverse for the 3'. And the hypervariable region, again known sequence, also will be learned with the polymerase reaction. So we will use these primers for forward here and for reverse here. And another point I want to mention is the Earth Microbiome project. This project aims for the collection of the microbiome variations on Earth in a library. Since the workload we're collecting all of these is very high, very high, everyone can contribute to this library project with the following protocol. They recommend the metadata file, the primers that can be used for hypervariable regions, and Illumina amplicon protocol are detailed on this guideline. It has two options, such as a single end and pair end for inserting the sequences into the programs. That's specific for the guidelines. These are all, again, detailed in the Earth Microbiome project protocols. For sequencing platforms, these are for the sequencing methods. These methods are differentiated depending on the size of the amplicon you want to use, depth of the sequence coverage, which is a rarity, I can also say that in another words, and the number of samples you have. These three things determines which method you are going to use. And currently using sequencing platforms are Illumina MiSeq, Illumina HiSeq, Ion Torrent, and Oxford Nanopore. What are these are basically, Illumina MiSeq is good for targeted sequencing. It can go up to 300 base pairs as paired, and this method is cost effective. It's basically the Illumina sequencing method. What is Illumina sequencing is like the primer that we are going to use attached with the adapters from the ends. Then these primers complemented to an array, and then in this array there are fluorescently tagged nucleotides, and these fluorescently tagged nucleotides matched with the polymerase reaction one by one, and each match they are shine. So with the all we are reading these shines in order to sequence them. This is how Illumina done. For Illumina HiSeq, this is also good for targeted sequence. It is actually the better quality version of Illumina MiSeq because it uses more strong lasers, and it can go up to 250 base pairs. For the ion torrent, this method detects the hydrogen ions released from the matching nucleotides. Every nucleotide, actually, when they match for transcription process, for polymerization, they release an hydrogen. So they basically detects these hydrogen ions for sequencing. And for Oxford Nanopore method, this is and also a new method that I can say that found by the Oxford University. In this method, a nanopore on the solid membrane surrounded by an electrolyte solution. The membrane split the solution into two, and reading has been done with the voltage change. It's basically works with the voltage change. It is good for complete 16S rRNA sequencing. And also, I have put in a YouTube video that for the Illumina sequencing, the more detailed explanation. Okay, what is Qiime-2 analysis pipeline? I have been used the Qiime-2, but what is it? I mean, it is a program, and this is a pipeline that used for dealing with the amplicon sequencing data analysis. It's a wrapping program actually for a lot of plugins inside required for the analysis. It is used mostly for diversity analysis, and I have been used for diversity analysis also in my research. Advantages of using this Qiime-2 program is easy to use. It's explained with the figures and the animations, and for the wrapper tool option, it's actually basically I can give you an example for this one. We want to do an OTU picking, an ACV picking, or in another name. We can use uClust, CDHIS, BLAST, Modder. All these can be used in this program. And since these all have different languages between them, we cannot use everything together or with different with using the same program, but Qiime-2 does that actually. Qiime-2 communicate each of these plugins so that we can use them. Okay. This is how it is actually done as an overview for the pipeline. First, the 16S genes in the wanted region has been tagged with DNA barcodes to know which variant belongs to which sample. then sequence them in the Illumina machine, just right here, and then editing the sequences for the best quality format. After editing and quality filtering, by phylogenetic tree has been generated, and after generating a phylogenetic tree and detailed edited sequences, we have done our analyses. And this is a picture for the emperor, by the way. For these analyses consists principal component analyses, alpha and beta diversities, income abundance analyses, more. Okay. For this is the experiment I have been mentioned before. We test the hypothesis of whether the Parkinson's disease influences the fecal microbiome or the microbiome contributes to the PD. For the investigation, mice that have suspectable Parkinson's disease genotype and the wild type genotype mouses put in 6 cages as mixed. They are not differentiated here with the wild type, here with the susceptible genotype. These are all mixed. Then 3 of these cages, healthy fecal microbiomes have been transplanted. All of these mice have healthy plantation, and all of these 3 have Parkinson's disease fecal microbiome plantation. And then we observed these mice for 49 days, and we have time to time took samples from them for the analyses, and I have been actually analyzed them for microbiomes for 16S RNA hypervariable region 4 investigation for diversity changes or for 49 days completely. Okay, then this is the basic experiment have been done, experimental process, and we are going to do analysis process. And this figure shows the overall analysis of every in the pipe of the Qiime-2 pipeline. Okay, so first, I'm not going to go into details because I will also mention all of these. So I'm going to go step by step, and we are here in this step, raw sequences step. We need 2 files for importing. We need to import our files into the Qiime-2 program right now because we took our sequences, raw sequences from the Illumina machine. Okay, we have 2 files, sequences files and barcodes files. And for the next step, for importing these, we have to do a protocol. We have to follow a protocol. But first, I want to mention about the sequences file. This file is basically contains the raw sequences that came from the Illumina machine. They come as a multiplexed form, as you see in here. Multiplex form means that they are jumbled each other. They are actually sequenced them as mixed. So this is why its name is multiplex. And these files comes as a FASTQ files. FASTQ files means quality scores also found in these files, in these FASTA files. Now for the barcodes files, it's the files that contains the barcodes that tag to the amplicons. We tagged them. We tagged our sequences, and they 2 sequences file and barcodes file imported together in Qiime-2 program with Earth Microbiome Project protocol. And for our experiment, we have used single-end Earth Microbiome Project importing protocol, which I will mention later. And also we want a metadata file for mapping. It's used for the samples and sequences. It contains the determined condition information of the samples, such as temperature, humidity, genotype values. These categories also contain 2 types, which are categorical and numerical categories. These all can be done in Google Spreadsheets or Microsoft Excel, this metadata file. For the results of this diversity, this file map map the sequences for the which category has the most effective on what. Okay, this is an example of metadata file, and also the metadata file I have been used for my experiment, the example experiment, PD, Parkinson's disease mouse experiment. As we can see in here, we have barcodes, genotypes, which is a categorical. We have cage ID, which is another categorical category. Donor, categorical. Donor status, categorical. Genotype, categorical. Days post-translated is numerical. This is our columns for in our metadata. Okay. At this step, we imported our sequences as an artifact into Qiime-2 with merging barcodes by using EMP protocol. They have been together formed as an artifact in Qiime-2 program. Right now, we are going to demultiplex them. Demultiplexing step is outlined in this figure. We have more than one option according to our sequences that are single-end or contains both forward and reverse primers, and we do demultiplex them. Also, we import them specifically, as you see, single-end sequences, paired-end sequences. Okay, we can also use. Okay, right now we're going to use demox EMP single-end method, but we can also use cut adapt method also as an alternative. But we are not going to mention this one today. I'm going to mention this one. Okay. For next, we're going to demultiplex our sequences. And we use the demultiplex for the demultiplex sequences, demux plugin, as I've shown here, EMP single in the code, which is detailed in the project, in the report. Okay. For demultiplexing, after the demultiplexing step, we are getting a quality score figure like this. These sequences from the multiplexed has been demultiplexed. We get the quality scores for our sequences. In this graph, we investigate the qualities, and we want them to be more than 20 at least for good quality. This is every one of these mentioning about the quality of the reading sequences, how it is read in the machine actually. So we can here, we want them to be higher than 20, which is here, and we all here have higher than 30. 30 is great, by the way. So we have a great example for our experiment. So we do not have to trim or truncate them from ends. Also, it is important that we have 150 base pairs, which is should not be too short. Otherwise, it can cause chimeric readings. Okay. After that, after this step, we are here. This is the step for the amplicon sequence variant picking method, which is a denoising method, also another name. There is more than one option again, but we're going to use data2. Why? These methods, the alternatives of our methods, has 3 options actually, more than 3 options in UNOIS 3. Okay, for data2 is the method we used. It is the most sensitive method for detection of the rare amplicon sequence variants. It uses lots of memory and time, but it is actually good for the use. And for the debuggler, for alternative of this, it limits the memory usage, but it decreases the sensitivity of the detection of the sequencing, detection of the ACV picking filtering. Actually, it takes less time, but it is decreased with the decreased sensitivity. All of these together are open source method. What is open source method? Is the sequences first checked with the referencing for detection for the amplicon sequence variants. If not found in the reference database, then looked at the similarity of each other de novo. For if they have similarity 95%, then more than 95%, then they are counted as the same variant. And for closed referencing, on the other hand, only uses a database. They do not do this de novo picking method. Also, you can use de novo picking too, with using unoise tree, and is the method for the alternatives. If it using closed referencing, open referencing, also de novo. Okay. After after denoising step, we have been used the data2 method for my experiment. I had time. Then I had 3 files. One of them is the denoising stats as a result files for the after the code. It shows how many sequences left and lost while the quality filtering process. It filters the sequences from non-chimeric ones. It actually follows like this, and this is the end product. And this is the non-chimeric ones, the end as can be seen. These are the sequence counts for each variant, each sample. Here, day 14, recipient 539, whatsoever. We can eliminate them with filtering. And if we found them, the quality sequence counts are low, we can eliminate them later because we want high quality. Another file we had this is the feature table for frequency table. It shows the frequency of the sequence variants of the samples. We want high as possible again. And also, we're going to use this medium frequency value for sequence depth determination with the alpha verification. Sequence depth determination has been done for the median frequency. This is used for the alpha verification sequencing method. And the other file, the last file we had, is the feature data for the sequences. This shows the sequence information of the variants. It can be obtained for each sequence variant found in all samples. Using the corresponding fastest sequence that we see here for each feature ID, we can validate them with the blast in the NCBI, which variant is found and is it correct or not. Okay, after getting this one, I generated a phylogenetic tree for the diversity analysis. For the generation of the phylogenetic tree, multiple sequence alignment of all sequences has been performed with the MUFT plugin using in the code, and filtered them together. Filtered them multiple sequence aligned sequences, and then we have been generated with using these multiple alignments and unrooted tree for the similarity between them with using the fast tree plugin. And after that, we have generated a midpoint for the conversion of unrooted tree to rooted tree again in the code with using the Qiime-2. Okay. And then the end product is an artifact of the phylogenetic tree. Okay, this pipeline figure shows the process of generation of the phylogenetic tree in code-wise, what has been done. In this figure, we can see the alternatives also present, but we followed this pipeline this way. Actually, we went to here, we filtered here, we aligned here, we filtered here, then we used fast tree, and we have generated an unrooted tree. After that, we midpointed them, and then we get here phylogenetic tree. Okay. After generation of phylogenetic tree, we want and feature table. Our goal is to making these is the core metrics results. We want to do analysis because of them also. So I will obtain the alpha, beta, and PCA and the Emperor 3D clustering analysis with this plugin, this core metrics results plugin. I need feature table and phylogenetic tree for it, and for this code as a plug in the plugin, but I still need one thing more, which is the sampling depth parameter of my sequences. So in order to get that, this step for getting the sampling depth require critical thinking. For sampling depth, I investigated the feature table first for how many sequences I count, sequence count I have in my samples. I general thinking about 1,000 sequence count is good for you. However, the best method is for determining the sampling depth is the alpha verification. Alpha verification is basically looking at which depth feature variance occurred and which depth, how many samples you have lost. I mean, the sequences have been completed. Actually, the better words is how many sequence counts you have in every sequence for every sample. And in here, we use the medium frequency as a parameter for the code of obtaining the most accurate approximation, which I have been shown before is like 4,000, 3,000 between them or something. Here we can see the operational taxonomic unit, and we did the alpha verification, and this is the result of the alpha verification, one graphic. And this is, we can see here the operational taxonomic units, which is the old name of amplicon sequence variance. Variance results at which depth a new variant is stopped to be can be seen in here. And this graph differentiates our sequences based on their genotype and of the mices, and can be seen that the observed OTUs has been stopped between 2000 and 2500 or something like that, but most observed until the 500 sequence count. And for our another graph where alpha verification shows that which depth samples have been lost. Here we can see that at 2000 depth we only lost one sample until this value. Therefore, 2000 is a good candidate for us. Also, we want to lose lowest sample count, lowest sample number, which is 1. We could have used 2500, but we would have lost too many samples. We want them to be high for the better quality, by the way, again. And in the feature table, it can be seen which sample have how many sequences counts. In here, we can see that this sample has lower than 2000, so it has lost, which is day 49, healthy cells, wild type. Okay. Using the feature table, phylogenetic tree, and sampling depth, core metrics result plugin have been processed. And this is the core metrics result folder as a result of the plugin, which is what I wanted for the whole thing. We obtained these files inside the folder, the vector QZA files like this one for the alpha diversity, and distance matrix is just like the here QZA file for the beta diversity files. For alpha diversity and beta diversity, we can see that every vector or distant matrices have been used for different purposes. For example, Fate's phylogenetic diversity in here is used for measuring the qualitative community richness with phylogenetic relationship of the features. For beta diversity, again, the Jacquard distance matrix, as an example, is used for measuring the qualitative community dissimilarity. Others are also used for other purposes, and I have actually investigated all of them. Okay, why we use the alpha diversity? What is the difference between the beta diversity from the alpha diversity between them? So alpha diversity is used for measuring the level of diversity within the samples, not between the samples. Beta diversity is measuring the dissimilarity between the samples. For example, in my genotypically suspectable Parkinson's disease samples, they can be measured for diversity, and I can do it with alpha diversity. But if I want to measure my sample dissimilarity between the wild type genotype and the Parkinson's susceptible genotype samples, it is beta diversity. So it's basically like this. For alpha diversity here, we used an high H test, Crookes-Wallace test has been used. It's basically the one-way non-parametric ANOVA test for bioinformatic analysis. It is used for meaning the groups, comparing the means of them, and more than 3 for each other groups. The alpha diversity answers the question of what is there and how much is there. And here we can see the richness comparison for alpha diversity samples of the samples specifically for genotype and donor type. It can be seen here that wild type genotype and healthy donor type samples have the highest community richness according to Fate phylogenetic diversity analysis. Also, the lowest susceptible genotype and PD donor, which is here what I mentioned, and this is what I mentioned for the highest one. This shows the richness of the highest when the healthier. Wild type and healthy, susceptible and PD. Okay, and this one here, the community evenness investigated. Evenness means that I'm gonna give an example for it. If I have 80 fruits in a basket, if 40 of them are apples and 40 of them are bananas, then we have a perfect evenness, which is the value of 1. It is basically the equality of the community groups. It has been looked for if dominance of species of the microbiome species is there or not. It can be seen in here in this graph. In here, dominance of a species is present in the susceptible PD samples, susceptible and PD samples, which is lower than others. The interesting clue here is that in the genotypically susceptible and healthy donors, evenness is the highest. In here, comparing with the wild type and healthy, it can be seen that it's even more even. Therefore, the genotype plays a role for the more even microbial diversity. Looking at the donor type, one does give you lower evenness more, which means that PD microbiome is effective on dominance of a species on the microbiome. Healthy and PD, wild type, wild type. For the beta diversity, I already mentioned the difference between the alpha. The beta diversity measures the level of the diversity between the samples from each other. It uses the Permanova, is used for testing the difference. The distance matrices have been used for that. It answers how similar or different are samples between each other. And for each distance matrices, one metadata column has been investigated. And here we see unweighted univariate distance matrix results of the samples for the beta diversity analysis. Unweighted univariate is used for measuring the qualitative community dissimilarity I shown before with accounting the phylogenetic tree. It can be concluded from these figures, all of these figures, that major role in the sample diversity has been sustained by the donor type rather than a genotype. And we investigated here that in the figures, as we look up closer, it can be also seen that donor type is way much effective for dissimilarity. In here, susceptible and PD, wild type and PD, here no low difference, but susceptible and PD, and susceptible and healthy, there is a high difference. So we see that this is caused by the donor type rather than genotype. However, we also have a minor genotypic effect also between them. And for the effectiveness of genotype and donor type of the sample diversity, how much they have been effective on diversities, our two tests also have been done in Qiime-2 with using Qiime-2. It can be concluded from the test that donor type is effective on diversity 30%, which can be seen in here. Donor is 30% and genotype is effective on 4% or 4.4%, 4.5% or something. The dominance, the major effect is by the donor, but genotype also have a minor effect. And in this figure, the pipeline for the alpha and beta diversity can be seen. What have been done is basically the pipeline is followed and the experimental values have been obtained. And in this figure, by the way, the beta significance, which is here beta group significance and alpha correlation, have been also done in my report in another experiment in order to correlate the metadata columns of the samples, whether they are related or not, how much related are they between each other. The metadata columns, just like humidity and the temperature, are they related to each other or not? It is used for that, and we followed this pipeline, by the way, as you see here. And also in here we see an emperor plotting for the visualization and PCOA results. For that, we see a 3D clustering has been taken from the emperor visualization. Colors have been labeled as genotype and donor type together. Here, red, blue, orange, and green. This is the PCOA analysis visualized by the emperor. And the clustering, it can be concluded from this image that mainly the samples clustered mainly due to their donor types. In here we have the PD donor type, and here we can see the healthy donor types. They are also clustered due to genotype between them. However, it's minimal, as seen in the green and blue. Also, susceptible and healthy increases in this one axis tree relative to time. Yet, wild type and healthy does not. They are more shallow. It is also proof of the genotype effect. Relative time may not be presented in this figure, But there's an animation perspective in the program also for the investigation on this one also. That is how I clearly state this this access tree thing. Okay, after we obtained the core metrics results, we clustered them, our samples, and looked for their diversity values. Now it is the time for the what we are looking for exactly. To do this, we classified every sequence variant in the microbiome that have been found by the taxonomic classification. We are going to name of our features actually. There are three types of classification methods: consensus blast, consensus research, are the alignment-based classification methods, basically end-top heads used for classification referred to a database. And the third option in here is the sklearn option. sklearn method is differentiated based on the machine learning classification. For this classification, the method you need a trained classifier. This method is used in my experiment, PD experiment, by the way. Okay. How are we going to train a classifier if this is the most sensitive method? In order to do that, for a specific hypervariable region taxonomic assignment, we need that database taxonomic text file. We will refer to the and the primers for the region we're looking at. What I mean is we need a database and we need the primers for the specific region we are interested, which is V4. There are databases for 16S, 18S, rRNAs, and ITS DNA. The green genes is 16S rRNA, and Silva is 16S, 18S, and Unite is the ITS gene, by the way. These are the databases. And then, in order to train a classifier, in order to train a classifier in the naive Bayes format for my experiment, I have been used the green genes 13/8 version, but not completely. It is 85% because OTU dataset, which does not contain all of the taxonomy of 16S, because it requires for complete training classifier needs a lot of RAM memory of my computer, so I do not have that much. So I used a portion of the database, and I have been tested also for the specific region in the report. in this one, in here, we can see the pipeline of the taxonomic assignment. There are lots of alternatives, as you can see, but we use this one, sklearn, and we will right now use our classifier and featured data to obtain the taxonomy assigned sequence variance results. And I have done that with the coding part, and this is the result of it. In here, it can be seen the phylum level taxonomic classification of sequence variants in our samples. They are separated based on the donor and genotype together, wild type and PD and etc. What we can conclude from this classification is dominant phylum is Bacteroidetes, which is the Bacteroidetes is the gray one. Then the Firmicutes is the brown one. And the second, PD donor samples without difference of the genotype only have 3 phyla, which can be seen in here that only 3 phyla, purple, brown, gray, purple, brown, gray. These are the PD samples, PD donor types. We can see more than 3 on the healthy ones. In healthy donors, yellow in healthy donors, yellow ones, which is Actinobacteria, also have been seen. Here you can see we have been seen healthy donors, and also susceptible and healthy ones include very blue ones, which is Microbial in here. These are interesting results because we have more than 3 in healthy ones and only 3 in PD ones. And we also have the blue ones and the yellow ones not together. And we are gonna look at why right now. And for investigation of this, we are suspected, actually I have been suspected for the cage specificity for the microbiome, uh, for the different microbiomes, because sometimes in the cages mice can gather up specific microbiomes to that cage. And as we look at the cage ID specific taxonomic assignment, we can see interesting results. Blue ones and the yellow ones are specific for some cages. And we conclude from these results that cage 35 and cage 31 have blue ones. And cage 31, 35, and 42 have the healthy donors. We concluded with the comparison with this one. And after that, the conclusion of these ones, yellow one, Actinobacteria does not shown in the PD donor samples without the difference of the genotype. Blue ones has only seen in susceptible genotype and healthy donors, which could be case specific phylum. Also minorly gray one, which is here is the Tenecricuties, also shown in cage 42, which can be also again case specific. And this is what we understand from our classification, but we need further investigation. So we have been, for the abundance analysis, so we have been used the ANCOM, which is the volcano plot method used. This is used for the abundance analysis of the genotype. This figure is for the genotype only and shown on the volcano plot. It can be seen that only 3 features here have the highest difference as in the abundance. This means that they're abundantly different between the genotypes compared to the others. In this graph, we can see that there is a 0 point, and this 0 point left side contributes to one genotype, the right side contributes the other genotype. And here we can see them more clearly. Here, for validation of the volcano plot, reject null hypothesis test results have been investigated also with this file. And in the figure 3 variants we have been seen, and these are the figures. These are the figures that have been rejected. The null hypothesis means they are different significantly among the genotypes, and this is for the showing the difference, how much different are they from each other. We can see here this feature is 101 to 2, but in wild type ones, but in susceptible none. In susceptible, none. In wild type, yes. In wild type, a lot, but after that, a lot in susceptible. In the beginning, not that much. This is what we are investigating. And this one have been done also for the donor types. Donor dependence analyses for donor only. In this one, we have more significant distribution on the volcano plot as V-shaped. Also in the reject null hypothesis test in here, we can see here more than 3, which is 24 feature variants have rejected the null hypothesis. This also validates that donor type has a major effect on abundance and levels of the feature variants also. compared to the genotype. And in this figure, we can see this the reject null hypothesis variance for the genotype and donotype interestingly differed from each other. How we conclude them, we compared these two together, and we resulted that these differences are this different feature variant names. I mean, these things are different in genotype and the donor type. So that means that genotype also has its own unique effect on the microbial abundance levels on specific features separated from the donor type. But majorly donor type has the major effect, dominant effect. And the variants that have reject null hypothesis for genotype and donor type investigated again. But after that, there's an interesting thing has been done. I have used the taxonomic assignment with using the random forest classification method for obtaining the collapsed fixed sequence variance at the species level, for using the abundance taxonomic analysis for the donor only, by the way. This is for the validation of the test is done properly or not. And it's investigated again that for the looking at the figures, the abundance difference levels for the taxonomic collapsed and contrast Firmicutes phylum Blauti as genus has the most abundance difference, which is here, this one, compared to the other one, all of the other one. And also the actinobacteria, which was the yellow ones in the graphical values, has the difference can be seen in the healthy donors, which can be seen in here. It is only found in the healthy ones, not the PD ones. In this slide, time relative distance difference analysis of the genotype and donor type together has been investigated. Another name of this one is longitudinal analysis. The figure of the volatility control chart shows the difference relative to the time. This is the 0 and this is the 49 days, basically. And these for the wild type healthy, susceptible PD, wild type PD altogether. And conclusion of this analysis seen that donor type has the major differentiator again for the shaping the microbial community, but also separated the variants again for the relative to time. For further investigation, Emperor has a specific unique attribute for the animational perspective for the analysis. Every mouse can be investigated in this one relative to time. In this one, every mouse has been colored in the different colors and animated according to relative to time, how different they are. It's concluded from this analysis that the healthy donor samples increased zinc on the axis tree, which I have been mentioned before, but in the PD samples, they have been changed more shallow. Here you can see axis tree, axis tree, axis tree. I'm sorry, axis tree. In this slide, the last analysis is projected residuals have been investigated. It is an analysis for a relative distance change for genotypic effect and donor type effect. It can be seen in these figures that donor type has a major effect for the differencing them in the scatter plot as they post transplant. In the genotype, it has a minor effect, but you can see they are differentiated a lot. But in the genotype, they also differentiated, but minorly. And this is another projected residual, which is relative to distances measures. We can see in here that also the genotype again has less effectiveness, and the donor type has more effectiveness. This is the genotype 1, which is more jumbled together each other, but this is a donor which is separated more each other, which we conclude from this one. The major effect on this one is here, donor type again. And in this one, in here, the lastly, for the all conclusion of the all analyses summed up in this slide for the whole experiment, for the whole example experiment of the PD. First, we investigated alpha diversity, and it showed that the donor type caused the major difference within the samples, and the genotype is weak. And then the beta diversity also showed the same result. While investigating PCA and Pearl plots, donor type again had the major difference caused. However, the genotype also had the minor differences with the cluster formation. And then we did a taxonomic classification, taxonomic assignment analysis, and it showed that the actinobacteria, which was the yellow one, phylum, is only seen in the healthy donor cells without that difference in the genotype, without differing the genotype. Also, the blue ones, the blue phylum Actinobacterium Microbial, phylum presence considered as a cage-specific phylum, because they only have been seen in cage 31 and cage 35. ANCOM results showed, the abundance analysis results showed the genotype has a unique effect on the changing difference of the abundances in the specific feature variants. And also, donor type abundance difference on amplicans and clearance variants had the major effect, but separated from the genotype. And lastly, the time relative analyses, longitudinal analysis, showed the samples with different donor types significantly changed relative to time. However, the genotype different samples also slightly change. Therefore, our hypothesis can be shaped as majorly Parkinson's disease influences the microbial community. However, genotypic influence of the Parkinson's disease influence the microbiome contributes to the development of Parkinson's disease, with separated from the microbial contribution variants. And this is all of my all of the presentation, and thank you for listening.